In the deepest oceans, darkest forests, and most hostile deserts, evolution has crafted nightmares and miracles, creatures that defy death, plants that cheat time, organisms so strange they rewrite the rules of life itself. This isn't horror. This isn't fantasy. This is Earthend. It's far weirder than you think. Close your eyes. Picture a creature that never dies, a plant that sleeps for years, a silent predator that glows in the dark. Now, open them. These aren't dreams. They're real. They're all around us, and they're proof that reality is stranger than any fantasy. Let's drift into the quiet, incredible world of Earth's most mysterious life forms. Imagine a world where death is optional, where creatures laugh in the face of extreme conditions that would instantly kill most life forms. This isn't the premise of a science fiction novel. It's the reality we live in, a planet teeming with organisms so strange they might as well be aliens. From the microscopic tardigrade that survives the vacuum of space to the leafy sea dragon that vanishes into underwater forests like a living ghost. Earth's biodiversity includes marvels that challenge everything we thought we knew about life. These biological oddities aren't just cabinet of curiosities, novelties. They represent evolution at its most creative. Solutions so bizarre they seem impossible until you witness them firsthand. Take the resurrection plant, for instance, a dried-up tumbleweed that looks completely dead, but springs back to vibrant green life after years of dormancy with just a few drops of water. Or consider the sea cucumber, which responds to predators by violently expelling its own internal organs, and then casually grows them back like nothing happened. These organisms don't just survive. They redefine what survival means, employing strategies so radical they make conventional biology textbooks seem hopelessly outdated. What's most astonishing is how these extremophiles and biological outliers aren't confined to some distant, inhospitable corner of the planet. They're all around us, in our forests, our oceans, even in our backyards. The more scientists look, the more they discover that life thrives in places and in ways we never imagined possible. It's the discovery of microbes living in boiling acidic springs, or worms thriving in ice, forces us to reconsider the very definition of habitable. These organisms don't merely endure their environments. They've transformed adversity into opportunity, evolving adaptations that border on the supernatural. This journey through nature's most eccentric creations isn't just about marveling at curiosities. It's about understanding life's incredible resilience and inventiveness. Every organism we'll explore is a testament to billions of years of evolutionary experimentation, of trial and error that has produced solutions both beautiful and grotesque. As we peel back the layers of their biology, we don't just learn about strange creatures. We gain profound insights into the fundamental principles that govern all life on Earth and perhaps beyond. The implications ripple outward, influencing fields from medicine to robotics to the search for extraterrestrial life. By studying these biological rebels, these rule breakers of the natural world, we're forced to ask, if life can do this here, what might be possible elsewhere in the universe? The tardigrade's resistance to radiation suggests organisms might survive interstellar travel. The respiration of deep-sea vent worms hints at how life could exist in the oceans of Europa or Enceladus. Each strange adaptation we uncover doesn't just expand our knowledge of Earth's biodiversity, it expands our imagination for what life could be. Prepare to have your perception of biology turned inside out. The creatures we're about to meet will challenge your assumptions, dazzle your senses, and perhaps most importantly, remind you that reality is always stranger than fiction. This isn't just science. This is nature showing off. If evolution had a hardcore survival competition, tardigrades would take home every trophy. These microscopic, 8-legged creatures, no larger than a speck of dust, boast survival skills that put even the most rugged wilderness experts to shame. Nicknamed water bears for their lumbering, bear-like gait under a microscope. Tardigrades are nature's unkillable champions, creatures that can weather the most extreme conditions the universe can throw at them. Scientists have subjected tardigrades to unimaginable trials, boiling them at 300 degrees Fahrenheit, freezing them near absolute zero, blasting them with radiation doses thousands of times lethal to humans, and even launching them into the vacuum of space. Yet, through it all, these millimeter-sized marvels simply shrugged it off. Their secret: a biological superpower called cryptobiosis, where they retract their legs, curl into a barrel shape called a ton, and essentially suspend animation by removing nearly all water from their bodies while producing special protective proteins. In this death-like state, their metabolism drops to 0.01% of normal activity, allowing them to survive for decades, perhaps even longer, until conditions improve. But tardigrades aren't just fascinating scientific oddities. Their biology is revolutionizing medical research. The same proteins that protect their cells from radiation damage could one day help shield astronauts on Mars missions or improve cancer radiation treatments. Their ability to survive extreme dehydration may lead to breakthroughs in preserving transplant organs. Even space agencies study them to understand the limits of life in the cosmos. These tiny creatures represent one of nature's most powerful lessons, that resilience isn't just about brute strength, but about biological ingenuity at its finest. Far from being the exception, tardigrades are everywhere, living in moss cushions, in your backyard, hiding in lichen on city rooftops, and inhabiting ecosystems from Himalayan peaks to deep-sea trenches. They remind us that superpowers don't just exist in comic books. Sometimes they're crawling through common moss under our feet, waiting for scientists to uncover their next survival secret. Their universal presence suggests that whatever catastrophe might befall Earth, the tardigrades will likely outlive us all as nature's ultimate emergency contingency plan, while tardigrades reign supreme in surviving extreme conditions. They are far from alone in nature's hall of ultimate survivors. Across our planet, in its deepest trenches, most acidic pools, and most inhospitable environments, evolution has crafted organisms that push the very boundaries of what we consider possible for life. These biological marvels don't just survive. Under extreme conditions, they thrive, rewriting the rule book of biology with every discovery scientists make about them. The undisputed champion of biological immortality is the immortal jellyfish Torritopsis dohrnii. This small, unassuming creature, no larger than your pinky nail when fully grown, holds the key to what may be nature's closest approximation to eternal life. When faced with injury, illness, or starvation, this jellyfish performs what can only be described as biological alchemy through a process called transdifferentiation. Its specialized cells transform back into their earliest developmental forms, essentially rebooting its life cycle. Imagine if you could respond to aging or disease by reverting to an embryonic state. This is precisely what the immortal jellyfish does naturally. Scientists have observed individual specimens undergoing this transformation multiple times in laboratory conditions, suggesting these creatures could, theoretically, live indefinitely under the right circumstances. The implications for medical science are staggering. Studying their cellular transformation processes could revolutionize how we approach aging, Keisha, regeneration, and even cancer treatment in humans. Even more perplexing is Deinococcus radiodurans, a bacterial species that holds the title for being the most radiation-resistant organism ever discovered. This microbe's earned the nickname Conan the bacterium for good reason. It can survive radiation, doses up to 5,000 grays, a unit of radiation absorption, while a mere 5 grays would be lethal to humans, and 1,000 grays would sterilize a laboratory's worth of equipment. When blasted with radiation that should shred its DNA to molecular confetti, Deinonychus simply pieces its genome back together with near-perfect accuracy within hours. NASA has taken particular interest in this extremophile, as its radiation-resistant properties could be key to developing microbial life support systems for space travel or even terraforming other planets. Some scientists speculate that organisms like Deinonychus could survive interplanetary travel, possibly explaining theories of panspermia, the idea that life can spread between planets, aboard meteorites, venturing into Earth's most extreme underwater environments. Reveals the Pompeii worm, Alvanella pompeiana, which makes its home in one of the planet's most hostile habitats, deep-sea hydrothermal vents. These worms thrive where temperatures reach 176 degrees Fahrenheit, 80 degrees Celsius, just shy of boiling point, all while enduring pressures that would crush most submarines and swimming through plumes of sulfur-rich chemicals that would be instantly lethal to nearly all other life forms. Their existence challenges everything we know about the temperature limits of complex organisms, as their proteins and cellular structures somehow maintain structural integrity at temperatures that should cause them to denature and break down immediately. The worms have developed a symbiotic relationship with heat-loving bacteria that form a protective fur along their backs, demonstrating nature's incredible ability to foster cooperative survival strategies under the most improbable circumstances. Beyond these famous examples, our planet hosts countless other organisms that defy biological expectations in equally astonishing ways. The Arctic wood frog Rana sylvatica survives being frozen solid during winter months by flooding its cells with glucose to prevent ice crystal formation, essentially turning itself into a froxicle for months at a time before thawing back to life in spring. The Sahara Desert ant Cataclyphus pickler navigates its blistering habitat by using polarized sunlight patterns combined with a sophisticated counting mechanism that tracks its steps, allowing it to venture far from its nest in search of food, while surviving surface temperatures that reach 140 degrees Fahrenheit, 60 degrees Celsius. The red flatbark beetle Cucugus clavops produces antifreeze proteins that allow it to survive temperatures as low as -150 degrees Fahrenheit, -100 degrees Celsius, making it one of the few complex organisms that can endure the winters of the Arctic Circle. Perhaps most mind-bending of all is the discovery of organisms that challenge our very definition of life itself. Viruses, existing in a shadowy realm between living and non-living, demonstrate that the boundary between biology and chemistry is far blurrier than we once thought. Then there are the indolents, microorganisms living deep inside rocks, sometimes kilometers below Earth's surface, surviving on nothing but the barest trickle of chemical energy, reproducing maybe once every 100 years, completely divorced from the sun-powered world we consider normal. Their existence suggests that life might be far more widespread in the universe than we imagine, possibly thriving in the subsurface oceans of Jupiter's moon, Europa, or the permafrost of Mars. The study of these impossible survivors is yielding transformative applications across multiple scientific disciplines. Medical researchers are looking to extremophile enzymes to develop more stable pharmaceuticals and vaccines that don't require refrigeration, a crucial need for developing nations. Material scientists are studying extremophile proteins to create stronger, more durable synthetic materials. NASA uses extremophile research to formulate planetary protection protocols and develop astrobiological detection equipment for future missions. Even the computing industry is taking notice, with some researchers investigating whether DNA storage systems based on extremophile genetics could create hard drives that last for millennia. Yet beyond their practical applications, these organisms force us to fundamentally reconsider the boundaries between the possible and impossible in biology. Each new discovery reveals that life is far more adaptable, more resilient, and more ingenious than we previously imagined. They demonstrate that where we see hostile, uninhabitable environments, evolution sees opportunity. In doing so, they expand our understanding of where life might exist, not just on our planet, but across the cosmos. As we continue to push the boundaries of human exploration, from the deepest oceans to other planets, these impossible survivors serve as both inspiration and guide, reminding us that life, in its infinite creativity, will always find a way. While evolution has produced astonishing survivors, it has also crafted master builders organisms that construct elaborate structures with mathematical precision, durable materials rivaling human engineering, and intricate designs that have baffled scientists for centuries. These are not simple nests or burrows, but sprawling, specialized architectures serving purposes so advanced they border on artistry. From the subterranean fungal skyscrapers of Tardigrades mushrooms to the colossal coral cities built by millimeter-long polyps, nature's architects have been perfecting their crafts for hundreds of millions of years, long before humans erected their first mud brick hut at the heart of the Amazon rainforest. An inconspicuous architect works in near darkness, the parasol ant. These tiny insects construct underground fungal farms spanning hundreds of square meters, some networks exceeding the volume of a 4-story building when scaled to human size. Worker ants meticulously cultivate their fungal crops, employing ventilation shafts that maintain perfect humidity and temperature. They even create waste chambers to prevent contamination, displaying an understanding of sanitation that matches human agriculture, the most breathtaking feature. These ants align their nest entrances using Earth's magnetic field to optimize airflow navigation skill. We've only replicated with modern compasses. Matching their ambition are the termites of Namibia's deserts, builders of towering cathedral mounds that stretch up to 5 meters high. These earthen spires are more than just dirt piles. They're climate-controlled megacities. Intricate tunnel systems act as natural air conditioning, with underground chambers absorbing moisture at night and vents channeling cool air during scorching days. Researchers discovered that the mound's north-south orientation minimizes heat exposure, maintaining an interior temperature of 87 degrees Fahrenheit, 31 degrees Celsius, year-round, regardless of whether it's 120 degrees Fahrenheit, 49 degrees Celsius, or freezing outside. Such precision has inspired architects to design energy-efficient buildings mimicking termite engineering, like Zimbabwe's Eastgate Center, which uses 90% less power than conventional structures. Beneath the ocean's surface, an even grander construction project unfolds. Coral polyps animals, no larger than a rice grain, secretly cement their skeletons into sprawling reefs over millennia. Australia's Great Barrier Reef alone comprises nearly 3,000 individual reefs and 900 islands, making it the only living structure visible from space. These tiny architects build with calcium carbonate, creating formations so sturdy they withstand hurricanes, yet so intricate that nanotechnologists study their microstructure for self-healing materials. The reef's limestone branches serve as underwater metropolises, supporting over 25% of marine species, despite covering less than 1% of the ocean floor. Even more mystifying is the geometric genius of honeybees. Their wax combs, built with millimeter precision, follow the honeycomb conjecture, a mathematical principle stating that hexagonal tiles use the least material to hold the most weight. It took mathematicians until 1999 to officially prove what bees have known for 130 million years, that hexagons are nature's perfect building blocks. The hives are so thermally efficient that NASA studied them for spacecraft insulation, while materials engineers mimic their wax for lightweight, ultra-strong composites used in everything from helmets to aircraft panels. But Complexity isn't limited to large structures. Microscopic diatom singles, single-celled, alginate, ornate silica. Shells in fractal-like patterns that no 3D printer can yet replicate. These glass shells are so precisely perforated that they manipulate light waves, inspiring new photonic technologies for fiber optics and solar panels. Similarly, the common spider spins silk stronger than steel by weight, with Darwin's bark spider producing webs stretching 25 meters across rivers, the largest of any land animal. Scientists now synthesize artificial spider silk for bulletproof vests and biodegradable sutures. Perhaps most astonishing is how these architects work without blueprints, foremen, or machinery. Their intelligence emerges from millions of years of evolutionary trial and error, encoded into instincts and simple chemical signals. A termite has no concept of thermodynamics, yet it builds climate-control towers. A coral polyp doesn't understand structural engineering, yet it erects earthquake-proof limestone cities. This phenomenon, known as stigmarchy, shows that decentralized systems can create extraordinary complexity. A lesson tech companies will apply to AI network designs. The implications stretch beyond biomimicry. If simple organisms can achieve such sophistication without consciousness, how does that reveal about the fundamental rules governing our universe? Why do hexagons and fractals recur from beehives to salt flats? How do blind creatures engineer ventilation systems that outsmart human HVAC apps? Each discovery peels back another layer of nature's hidden blueprints, ones that could revolutionize urban planning, material science, and even space colonization. The thermite mound's passive cooling, for instance, might help build self-regulating habitats on Mars, while synthetic diatoms could lead to solar cells that grow themselves. As we uncover these biological marvels, a humbling truth emerges. Many human innovations were perfected by other species eons ago. Concrete. Romans learned from volcanic ash, but corals have used biochemical cement for 500 million years. Skyscrapers. Termite mounds dwarf our structures relative to their builders' size. Waste recycling. Ant colonies operate at near-zero landfill waste. In an era of climate change and resource scarcity, these ancient architects hold survival secrets we've only begun to decode. Their structural genius, forged through eons of environmental pressures, may well be the key to our own sustainable future. The next time you see a beehive or anthill, pause and consider that humble structure represents an engineering legacy far older than the pyramids. It's crafted by creatures that never went to school, never read a textbook, yet mastered physics, chemistry, and urban design long before we existed. In their precise geometries and climate-adapted forms lies not just scientific wonder, but a message. The best solutions often come not from reinventing nature, but from finally understanding its timeless blueprints. Beneath the cacophony of the natural world thrives an ancient web of secret dialogues, chemical whispers, electrical pulses, exchanged between organisms with a sophistication that makes human language seem crude by comparison. These aren't chance interactions, but intricate communication systems refined over hundreds of millions of years, where a single misread signal can mean the difference between survival and oblivion. From the ultrasonic distress calls of plants to the complex grammar of dolphin whistles, life has developed a hidden lexicon far more nuanced than we once imagined, rewriting our understanding of consciousness, cooperation, and what it truly means to speak. The most ubiquitous language on Earth is chemical effect, demonstrated spectacularly by the acacia trees of the African savanna. When giraffes browse their leaves, the trees release ethylene gas that travels through the air at 1.5 miles per hour, warning nearby acacias to pump their foliage full of bitter tannins within minutes. Scientists discovered this arboreal SOS spreads up to 90 feet, creating an invisible fence that forces giraffes to feed upwind. Even more remarkably, certain acacia species have adapted to cry wolf, emitting false alarms when no giraffes are present, causing neighboring trees to waste energy on defense and giving the liar a competitive growth advantage. This botanical deception reveals vegetal communication isn't just reactive, but startlingly strategic. Beneath our feet, an even more astonishing linguistic network operates. Mycorrhizal fungi weave through soil in vast subterranean webs, called the Wood Wide Web, linking tree roots via fungal filaments thinner than human hair. Through these connections, trees trade carbon, nitrogen, and water with elder mother trees, even nurturing shaded seedlings by diverting nutrients through the fungal internet. Research by ecologist Suzanne Simmerd shows Douglas firs recognize their offspring, preferentially supplying them with extra resources while limiting support to unrelated saplings. The system operates via biochemical on-off signals analogous to computer code, with some fungal species acting as translators between different tree dialects. When loggers clear forests, surviving trees send distress chemicals through the network, triggering accelerated growth in remaining members of phenomenon. Scientists compared to neurological trauma responses in animals. The ocean conceals communication. Marvels just as profound. Coral reefs, often mislabeled as silent landscapes, actually crackle with noise, a phenomenon marine biologists term the reef orchestra, using hydrophones. Researchers identified that parrotfish grind limestone with beak-like jaws, producing audible crunching. Snapping shrimp create 200 decibel snaps with their claws louder than gunfire, while male damselfish generate pulsed songs by vibrating swim bladders to attract mates. Most remarkably, baby corals spend their larval stage floating in open water before locating home reefs by listening for this acoustic signature. A discovery forcing us to reconsider if colonies, separated by miles, might actually function as one interconnected superorganism. Airborne communicators achieve equal brilliance in their own mediums. Honeybees perform elaborate waggle dances, encoding precise GPS coordinates, the angle indicating direction relative to the sun, duration revealing distance, and vibrations conveying nectar quality. A 2022 study at the University of Tokyo proved bees adjust their dance movements based on peer feedback, essentially holding democratic debates about optimal foraging. Sites of form, swarm, intelligence surpassing early human tribal decision-making. Meanwhile, elephants communicate via seismic rumbles that travel 20 miles through ground vibrations, their footpads acting as biological seismographs to detect messages about distant waterholes or predators. Bioacousticians found matriarchs even use specific rumble patterns to name individual family members, and identifier system linguists compared to human proper nouns. Microbial speech proves the most alien yet revolutionary. The bacteria Pseudomonas aeruginosa organizes group behaviors through quorum sensing, releasing molecular signals that count population density like a microbial census. At critical mass, they simultaneously launch coordinated attacks, forming antibiotic-resistant biofilms in human lungs. Doctors now disrupt these chemical conversations with quorum quenching drugs that jam bacterial signals, a breakthrough leading to next-generation infection treatments. Even viruses communicate. Phage viruses leave peptide messages inside infected cells to guide subsequent viral generations. Discovery rewriting virology textbooks, the implications cascade across disciplines. Deciphering prairie dog sentences which distinguish between coyotes, hogs, and even human shirt colors revolutionizes animal cognition studies. Analyzing dolphins' signature whistles, functionally equivalent to names, suggests self-awareness levels nearing human children. Forest communication networks inspire decentralized AI, language models that require no central processor, with tech firms already patenting mycorrhizal-inspired first systems that distribute data like tree roots, share nutrients. Yet, amid these revelations, existential questions loom. If a tree warns its kin of danger via chemical screams, do plants feel pain? When bees debate optimal routes via dance democracy, where does instinct end and cognition begin? As we peel back layers of nature's linguistic complexity, we're forced to confront our biased definitions of intelligence, awareness, and even language itself. Perhaps the greatest lesson lies not in the complexity, but in the purpose of these exchanges. Whether fungal chemical pulses or whale song sonatas, every natural dialogue serves one unifying function: sustaining life through shared knowledge. In our age of fractured human communication, nature's silent whispers remind us that true language's highest purpose isn't merely to transmit information, but to weave the invisible threads that hold ecosystems, ultimately our planet, together. Next time you walk through a forest, swim in the ocean, or watch bees flit between flowers, remember you're standing amid an ongoing conversation older than civilization itself. Its vocabulary written in chemical chains, electrical frequencies, and vibrating atoms. To hear it, we need only learn to listen, while we build laboratories to conduct experiments. Nature has been running the ultimate R&D department for billions of years, one whose innovations put our most advanced technologies to shame. From spiders spinning silk stronger than steel to mollusks crafting ceramics at water temperatures, organisms have perfected chemical processes that human scientists still struggle to replicate. These biological alchemists don't need supercomputers or particle accelerators. Their laboratories exist within their very cells, conducting molecular miracles we're only beginning to understand. Consider the common spider, nature's most underrated polymer scientist. Its coned golden silk orb weaver produces silk with such precise, tensile strength that Boeing studies its protein structure for next-generation aircraft materials. But the real marvel lies in the spider's spinnerets, microscopic nozzles that transform liquid protein to solid thread through a process combining pH changes, water removal, and mechanical shearing. Scientists attempting to mimic the spinning technique can't yet match its energy efficiency. Spiders produce silk at room temperature using only amino acids and water, while industrial fiber production requires toxic solvents and massive energy inputs. Even more remarkably, some spiders can tune their silk's properties by adjusting glandular secretions, creating stretchy capture spirals, while maintaining rigid support threads in the same web. Such precision puts our synthetic material science to shame. The ocean's master chemists achieve equally astonishing feats. Limpets, small sea snails clinging to tidal rocks, manufacture teeth made of goethite, an iron-based mineral arranged in fibers just 100 nanometers thick. These teeth rank as the strongest biological material ever discovered, outperforming Kevlar and even carbon fiber. At UC Riverside, engineers attempting to replicate limpet tooth formation discovered the snails control mineralization through specialized proteins that guide iron atoms into perfect crystalline alignment at ambient temperatures, a process we can only crudely imitate using high-temperature forges. Their dental formula could revolutionize everything from bone grafts to spacecraft shielding. Perhaps most humbling are nature's solutions to chemistry's eternal challenge, breaking down stubborn polymers. Despite our best efforts, plastic waste remains a global crisis. Yet the humble waxworm Galleria melanella, the bane of beekeepers, casually eats through polyethylene bags, using symbiotic gut bacteria that evolved to digest beeswax. Researchers at Spain's Ipatec Institute found these caterpillars' enzyme cocktails degrade plastic 1,400 times faster than standard industrial methods, working at room temperature without producing toxic byproducts. Similar plastic-eating bacteria have since been discovered in landfills and even the Great Pacific Garbage Patch, suggesting evolution has already begun solving pollution through microbial adaptation. Plant biochemists demonstrate equally sophisticated expertise. The lotus leaf's famous water repellency stems from nanostructured wax crystals that create microscopic air pockets a design principle now applied to self-cleaning fabrics and non-fogging eyewear. But new research reveals an even more startling capability when damaged. Lotus leaves regrow their waterproof coating by precisely depositing lipid molecules along geometrical patterns dictated by the plant's epidermal cells. This self-repair mechanism outperforms any human-made hydrophobic coating, maintaining perfect water-beating properties despite harsh weather and physical abrasion. Microbes push biochemical boundaries furthest. Bacteria breathe by transferring electrons to external minerals through conductive nanowires, essentially generating electricity from dirt. Engineers now deploy them in microbial fuel cells that clean wastewater while generating power. Other extremophiles, like Sulfolabus, archaea, thrive in volcanic hot springs by stabilizing their DNA with reverse twisting enzymes that could revolutionize PCR testing. Even viruses contribute. The T4 phage builds its capsid with geometric perfection, assembling 200 protein subunits into a 20-sided structure through what materials scientists call nature's perfect origami. The implications cascade across industries. Spider silk proteins enhance biodegradable medical sutures. Limpid tooth principles guide flexible ceramics for dental implants. Plastic heating enzymes inspire zero-waste recycling systems, mimicking waxworm digestion. Each discovery exposes the hubris of assuming human technology represents evolution's pinnacle, when, in truth, we're newcomers, reverse engineering solutions perfected over eons. Fundamentally, these organisms demonstrate that nature's chemistry operates by different rules than human laboratories, water-based instead of solvent-dependent ambient temperature, rather than energy-intensive self-assembling instead of externally manufactured waste. Cycles, opposed to toxic byproducts as synthetic biologists, harness these principles, programming bacteria to produce medicines or algae to secrete biofuels. We edge toward a future where biotechnology merges with sustainability. Perhaps the ultimate lesson lies not in copying nature's products, but in adopting its mindset that true innovation solves problems without creating new ones, and that every waste is merely an unused resource waiting for the right organism to transform it. The next time you see a spider spinning silk or a limpet clinging to a wave-battered rock, recognize that you're witnessing biochemistry's greatest masters at work. Lifeforms conducting nanoscale engineering with elegance. We're only beginning to comprehend. Their silent research continues, as it has for millennia, hidden in plain sight, waiting for us to learn its lessons in nature's relentless arms race between predator and prey. Some organisms have perfected the art of deception to such extraordinary degrees that the line between reality and illusion becomes indistinguishable. These are not simple color changes or fleeting tricks, but elaborate, layered performances refined over millions of years. Biological theater so convincing, it puts human stage magic to shame. From spiders that mimic ants with uncanny precision to orchids that perfume themselves into irresistible love traps, evolution has crafted the ultimate illusionists, each specializing in forms of trickery that reveal profound truths about perception and survival. Consider the ghost mantis Philocrania paradoxa, a cryptic insect that has elevated camouflage to an art form. Unlike typical mantises that blend into green foliage, this master of disguise mimics dead, decaying leaves down to the smallest detail. Its body features jagged extensions resembling leaf edges ravaged by insects, complete with decay spots of darkened pigmentation and veins running through its exoskeleton like desiccated foliage. When disturbed, it sways gently like a leaf caught in a breeze, a behavior called vegetative rocking that enhances the illusion. But the true brilliance lies in its adaptive coloration. Specimens from rainier regions develop more vibrant, fresh leaf greens, while those in arid zones turn crisp brown-yellow, demonstrating an environmental awareness that adapts their performance to local conditions. Entomologists confirm that predators spot ghost mantises 70% less frequently than other mantis species, a success rate that drops to zero when the insects remain motionless. The animal kingdom's most audacious impersonators, however, may be the mimic octopuses, the Octopus, mimicus of Indonesia's coral reefs. While all cephalopods can change color, this shapeshifter takes the skill to unprecedented levels by physically altering its body shape and movement patterns. Within seconds, it can morph into at least 15 different species, flattening its body to impersonate a flounder, raising 2 arms to mimic the undulating form of a sea snake, or clustering 6 arms together to resemble the spiraling shell of a hermit crab. Marine biologists discovered each imitation serves specific purposes. The sea snake act deters predatory damselfish, which feared the venomous reptiles, while the flounder disguise helps it scout open sand flats inconspicuously. Most remarkably, individual octopuses develop personalized repertoires based on local threats, selecting impersonations from what scientists describe as a cognitive library of regional species, proof that these mollusks learn and customize their deceit. Vegetable fraud reaches its pinnacle with orchids in the genus Ophrys, nicknamed bee orchids for their scandalous reproductive strategy. Their flowers don't merely resemble female bees, they replicate the insect's wing patterns, fur textures, and even pheromone signatures with chemical precision. Male bees attempting to mate with these floral imposters become unwitting pollen carriers, transferring the orchid's genetic material between plants in what botanists call pseudocopulation. The system depends on extraordinarily specific deception. Each Ophrys species targets particular bee species, evolving its lures to match slight variations in the pollinator's visual and chemical cues. Some tropical orchids take the ruse further by mimicking the appearance and scent of rotting meat, attracting carrion flies that typically seek out decomposing carcasses to lay eggs, only to leave pollen-coated rather than depositing their brood. Fungi employ biochemical trickery, equally sophisticated. The zombie ant fungus Ophiocordyceps unilateralis doesn't just kill its insect host, it hijacks the ant's central nervous system, forcing the victim to clamp onto vegetation at the perfect height and humidity for fungal growth. Autolycal studies revealed the fungus selectively releases compounds that override the ant's normal behaviors, while keeping essential systems functioning just long enough to complete its macabre performance. Mycologists recently discovered the fungus doesn't act alone. It depends on a suite of helper microbes that suppress the ant's immune system and assist with neural, manipulational, parasitic partnership, analogous to hackers collaborating to breach a security system. The evolutionary implications are profound. Elaborate deceptions suggest nature selects not just for physical traits, but for what cognitive scientists call theory of mind, the ability to model another organism's perceptions and exploit them, whether through visual mimicry, chemical forgery, or behavioral manipulation. The most successful deceiters demonstrate an uncanny understanding of their audience's psychology and sensory capabilities. Biotechnology now seeks to harness these principles, with researchers developing camouflage materials inspired by cephalopod skin and agricultural pheromones based on orchid bee scams. Yet, for all our imitation, we've barely scratched the surface of nature's deceptive genius, a realm where the best trick is making the impossible seem effortless, and where every illusion serves the most pragmatic purpose of all: survival. The natural world operates on a timescale incomprehensible to human experience, where evolutionary changes normally occur across millions of years. Yet scattered across our planet are organisms that seem to have stepped out of the fossil record entirely, their forms frozen in biological amber despite the passage of hundreds of millions of years. These living fossils represent some of the most successful designs evolution has ever produced. Life forms so perfectly adapted to their niches that they've resisted modification despite countless environmental upheavals. Their existence challenges our fundamental assumptions about evolutionary progress, survival strategies, and what it truly means to be primitive. The coelacanth remains perhaps the most celebrated of these prehistoric survivors. When marine biologists confirmed the capture of a live specimen off South Africa in 1938, nearly 70 million years after their supposed extinction, it amounted to one of the greatest zoological discoveries of the 20th century. These deep-water fish, now represented by two living species, Latimeria chalumnae and L. manedoensis, exhibit anatomical features virtually unchanged from 400-million-year-old Devonian fossils. Their most striking characteristic, paired lobe. Fins, containing bones, homologous to terrestrial animals' limbs, provides an unprecedented living window into vertebrate evolution's most pivotal transition, the move from water to land. Unlike any other living fish, slug can swim using an unusual alternating fin gait strikingly similar to tetrapod walking patterns, offering biologists unprecedented opportunities to study how locomotion evolved during this critical evolutionary leap. Yet, coelacanths are far from evolutionary failures. Their persistence demonstrates remarkable biological refinement. Contemporary research reveals these fish possess numerous specialized adaptations for their deep-sea twilight zone habitat between 150 to 250 meters depth. Their eyes contain a unique crystalline structure that amplifies dim bioluminescent light, while specialized electroreceptors called rostral organs help detect prey in near total darkness. Their metabolism operates at an extraordinarily slow pace to conserve energy in their cold, food-scarce environment. They gestate young for 5 years, the longest of any vertebrate, and may live up to a century. Their genomes show unusually slow mutation rates, with key developmental genes remaining stable across vast geological timescales, a phenomenon geneticists now believe could unlock secrets about slowing biological aging processes. Equally astonishing are horseshoe crabs, Limulidae, marine arthropods whose basic body plan has persisted for an estimated 450 million years, surviving all 5 mass extinction events. These blue-blooded creatures are neither true crabs nor crustaceans, but rather distant relatives of arachnids, their closest living kin being spiders and scorpions. Their alien physiology includes multiple primitive eyes, including to compound, lateral eyes and several rudimentary light sensors, 10 legs used for walking, swimming, and passing food to a centrally located mouth-long, rigid tailspine, telson, used for righting themselves when flipped blood, containing copper-based hemocyanin that turns electric blue when oxygenated. Most remarkably, horseshoe crab blood contains amoebocytes, producing limulus amoebocyte lysate, LA, a clotting agent uniquely sensitive to bacterial endotoxins that remains medically irreplaceable for contamination testing in vaccines, implants, and intravenous medications. The pharmaceutical industry harvests over 500,000 crabs annually in a sustainable bleeding process that returns them alive to the ocean after extraction, making them arguably the most valuable marine invertebrates pound for pound, even among plants. Ancient relics persist in unexpected corners. Until 1994, Wallimea pines, Wallimea nobilis, were known only from 200-million-year-old fossils until a park ranger stumbled upon a small stand of living trees in a remote Australian canyon. DNA analysis confirmed these conifers as genuine dinosaur trees, living representatives of an Araucaria lineage, thought extinct for 70 million years. Just 90 adult trees survived naturally, clustered in a single inaccessible gorge, where they grow into towering 40-meter giants with unique bubbling bark texture, unlike any other conifer double or triple tree trunks growing in perfect unison cones that eject all male pollen simultaneously in explosive bursts. Seedlings that can sprout from underground lignotubers after fires. Ecologists now believe their survival depended on fire-resistant bark and these rarely seen canyon microclimates, where conditions remained stable since the Cretaceous. Wollemi pine specimens have since been propagated worldwide in a Noah's Ark-style conservation effort, with botanical gardens serving as modern arks, preserving this prehistory among skyscrapers and city streets. At the microbial scale, stromatolites offer the most humbling encounter with deep time. These living rock formations, built by cyanobacteria colonies, still grow in Western Australia's Shark Bay and isolated volcanic pools, using the same layered accretion process as their ancestors employed 3.5 billion years ago. Modern stromatolites construct themselves through daily secretion of calcium carbonate, cement vertical, its growth measured in mere millimeters per year, photosynthetic biofilm layers, managing extreme salinity, internal microorganisms cooperating and fossilized harmony. The discovery that these still living structures match Earth's oldest fossils perfectly revolutionized our understanding of life's origins, proving photosynthetic microbes shaped our atmosphere billions of years before multicellular life arose. What unites these biological time travelers is an extraordinary lesson about evolution's true nature. Their persistence demonstrates that success isn't measured by complexity or diversification alone, but by resilience, the ability to find solutions flexible enough to weather deep time's vicissitudes, yet specialized enough to resist displacement by newcomers. In an era of accelerating biodiversity loss, studying these survivors becomes doubly urgent. They represent irreplaceable genetic libraries containing lessons about perseverance we desperately need to decode. Their genomes may hold clues about resisting climate shifts, surviving mass extinctions, adapting to environmental changes on planetary scales, knowledge potentially vital for preserving our own species' future. Perhaps most philosophically profound is what these organisms reveal about time itself. For them, the Mesozoic isn't ancient history, it's yesterday. Their bodies remain living archives of worlds long vanished, yet stubbornly refusing to disappear entirely. They force us to expand our temporal horizons beyond human scales and recognize that nature measures success not in years or centuries, but in geological epochs. In an age where humanity grapples with its own fragility, time-defying survivors whisper an alternative possibility, that endurance belongs not to the strongest or fastest, but to those who mastered the art of perfect adaptation. Their continued existence represents life's most profound magic trick, making permanence out of change itself. In the intricate tapestry of life, the most remarkable stories often unfold in the shadows, where organisms engage in profound partnerships that blur the lines between individuality and community. These symbiotic relationships are not mere happenstance tides. They're the result of millions of years of evolution, where species have learned to rely on one another for survival, growth, and even reproduction. The complexity of these interactions reveals a hidden world of cooperation that challenges our understanding of life itself. Take, for instance, the relationship between the clownfish and the sea anemone. The clownfish, with its vibrant orange and white stripes, finds refuge among the stinking tentacles of the anemone, which would deter most predators. In return, the clownfish provides the anemone with nutrients from its waste and helps keep it clean by removing debris and parasites. This mutualistic relationship is a dance of trust and dependence, where both parties thrive in an environment that would otherwise be hostile. The clownfish's ability to navigate the anemone's stinging cells is a marvel of adaptation, showcasing how evolution can shape behaviors that enhance survival. Similarly, the relationship between oxpeckers and large herbivores like rhinos and buffalo illustrates the intricate balance of nature's partnerships. Speckers perch on the backs of these massive animals, feeding on ticks, dead skin, and parasites. While the birds benefit from a steady food source, the herbivores enjoy a form of pest control. However, This relationship is not without its complexities. Some studies suggest that oxpeckers may also peck at their host's wounds, drawing blood and causing harm. This duality raises questions about the nature of symbiosis and the fine line between mutualism and parasitism. In the depths of the ocean, the relationship between coral polyps and zooxanthellae algae exemplifies the beauty of symbiosis. Coral reefs, often referred to as the rainforests of the sea, are built by tiny coral polyps that secrete calcium carbonate to form their skeletons. These polyps harbor zooxanthellae, photosynthetic algae that provide them with energy through the process of photosynthesis. In return, the coral offers a protected environment and the nutrients necessary for the algae's survival. This partnership is so vital that when environmental stressors, such as rising sea temperatures, disrupt the relationship, corals expel their algal partners in a phenomenon known as bleaching. The loss of this vibrant partnership can lead to the collapse of entire reef ecosystems, highlighting the fragility of these interconnected lives. Even in the soil beneath our feet, symbiotic relationships flourish. Mycorrhizal fungi form associations with the roots of most plants, extending their hyphae into the soil to absorb water and nutrients, particularly phosphorus. In exchange, the fungi receive carbohydrates produced by the plants through photosynthesis. This underground network not only enhances plant growth, but also fosters communication between different plant species, allowing them to share resources and warn each other of impending threats. The mycorrhizal network acts as a lifeline, connecting trees and plants in a vast web of life that sustains entire ecosystems. As we delve deeper into the world of symbiosis, we encounter the fascinating relationship between leafcutter ants and the fungi they cultivate. These industrious ants cut leaves and transport them back to their underground nests, where they use the plant material to grow a specific type of fungus. The ants feed on the fungus, which in turn relies on the ants for its growth and propagation. This remarkable partnership showcases the complexity of mutualism, as both species have evolved specialized traits to support one another. The ants have developed a sophisticated system of agriculture, complete with pest management and waste disposal, demonstrating that the roots of agriculture may lie far deeper in the evolutionary tree than previously thought. In the realm of microorganisms, the relationship between humans and their gut microbiota serves as a poignant reminder of the power of symbiosis. Our bodies host trillions of bacteria, fungi, and other microorganisms that play crucial roles in digestion, metabolism, and immune function. These microbial communities help break down complex carbohydrates, synthesize essential vitamins, and even influence our mood and behavior. The delicate balance of our gut microbiome is essential for our health, and disruptions to this ecosystem can lead to a range of health issues, from obesity to autoimmune diseases. This intimate partnership underscores the idea that we are not solitary beings, but rather a complex amalgamation of diverse life forms working together for mutual benefit. As we explore these intricate relationships, it becomes clear that symbiosis is not merely a survival strategy. It is a fundamental aspect of life on Earth. The interconnectedness of species fosters resilience, adaptability, and innovation, allowing ecosystems to thrive in the face of challenges. In a world increasingly defined by competition and individualism, nature's symbiotic partnerships remind us of the power of collaboration and the importance of nurturing our connections with one another and the environment. In the face of climate change, habitat destruction, and biodiversity loss, understanding and preserving these symbiotic relationships is more crucial than ever. As we strive to create sustainable solutions for our planet, we must look to nature's time-tested strategies for inspiration. By embracing the principles of cooperation and interdependence, we can forge a path toward a more harmonious existence, where both humans and the natural world can flourish together. The next time you encounter a clownfish darting among the tentacles of an anemone or witness the industrious leafcutter ants at work, take a moment to appreciate the intricate dance of life unfolding before you. These partnerships are not just fascinating biological phenomena. They are a testament to the resilience and creativity of life itself. In a world that often feels fragmented and divided, nature's symbiotic superorganisms offer a powerful reminder that we are all connected, and that our survival depends on the strength of our relationships with one another and the ecosystems we inhabit. Evolution is not a process of random chance, but a grand laboratory operating on planetary scales, one where trillions of organisms serve as both experimenters and subjects in nature's endless quest for survival. For nearly 4 billion years, life has conducted iterative trials on everything from protein structures to behavioral strategies, covering engineering solutions that routinely surpass human innovation in efficiency, sustainability, and elegance. These are not discrete adaptations, continuous experimental processes that proceed with both ruthless pragmatism and accidental brilliance. Each organism embodying a living testament to perseverance through trial and error on the kaleidoscopic reefs of Indonesia. The mimic octopus, the Octopus mimicus, serves as evolution's premier performance artist, conducting daily experiments in deception. Unlike fixed camouflage patterns, this cephalopod actively tests new impersonations against real-world threats, cataloging predator responses like a marine field researcher. Marine biologists have documented individuals cycling through at least 15 different animal impressions, from lionfish to sea snakes to stingrays, adapting their performances based on nearby predators' reactions. Some even appear to combine disguises, merging qualities of multiple species into composite illusions, never observed in nature. The octopus doesn't inherit this repertoire genetically, but develops it through observation and practice, proving that evolutionary experimentation occurs across both geological and individual timescales. Even more astonishing trials unfold inside ordinary acorns, where oak trees employ a reproductive strategy botanists call predator saturation, each mast year occurring every 2 to 5 years in synchrony across entire forests. The trees flood the ecosystem with millions of excess nuts, overwhelming seed predators through sheer numerical excess. This dynamic adjustment, controlled by hormonal responses to weather patterns and resource availability, demonstrates how plants conduct long-term ecological experiments spanning generations. The trees remember which strategies succeeded through epigenetic markers, chemical tags on DNA that activate or silence genes based on past experiences without altering the genetic code itself. Modern agriculture now apes these principles through polyculture farming techniques that mimic forest mast cycles to naturally control pests. The microbial world operates as Earth's most prolific R&D department, with bacteria exchanging genetic material through processes like horizontal gene transfer, effectively trading survival strategies in real time. In Japanese wastewater treatment plants, scientists discovered a novel bacterial strain, Ideonella sakaiensis, that had recently evolved the ability to digest polyethylene terephthalate, PET plastics. Genetic analysis revealed this breakthrough occurred when environmental pressures prompted bacteria to reshuffle enzyme-producing genes acquired from soil microbes, creating an entirely new biochemical pathway. Nature's experimental process here was neither slow nor random, but a rapid, targeted response to a man-made challenge, proof that evolution's laboratory recognizes human activity as just another variable to adapt to. Ant colonies engage in sophisticated field testing of transportation networks that puts urban planners to shame. Research on Argentine ants, Linopathema humo, shows they optimize foraging trails using principles identical to computer algorithms that manage internet traffic flow. When presented with multiple paths to food sources, the colony, collectively, identifies and reinforces the most efficient route through pheromone markers, essentially crowdsourcing route optimization through distributed experimentation. These decentralized systems have inspired ant colony optimization algorithms now used to streamline shipping routes, telecommunications networks, and even disaster evacuation planning. Perhaps the most profound experimental arena exists inside every cell. The immune systems of jawed vertebrates perform what scientists call somatic hypermutation, deliberately mutating antibody genes millions of times per second to stumble upon pathogen-countering proteins. This controlled self-experimentation occurs in specialized lymph node structures that function like microscopic testing facilities, where B cells undergo frantic genetic trial and error to discover effective immune responses. The successful designs then proliferate, while failures undergo programmed cell death, essentially the biological equivalent of rigorous product testing. Pharmaceutical companies now mimic this approach through directed evolution techniques to develop new drugs and vaccines. The experimental ingenuity of simpler organisms puts many human technologies to shame. Slime molds, Fusarium, polycephalum, lacking even a rudimentary nervous system, consistently solve complex maze puzzles and recreate optimized transport networks that match or exceed human-designed infrastructure. When presented with oat flakes, arranged to mimic Tokyo's rail system, the organism's branching growth patterns spontaneously constructed near-identical layouts to the actual subway, complete with redundant connections at key transit hubs. Materials scientists studying this behavior have developed self-organizing conductive gels that could lead to living electronic circuits capable of self-repair. Plants conduct their own botanical trials through epigenetic memory systems that remember environmental stressors across generations. Sagebrush plants attacked by herbivores release volatile compounds that warn neighboring vegetation to activate defensive chemicals, with the most effective warning signals being adopted as dominant responses across the population. Recent studies show descendant plants retain this chemical knowledge for multiple generations, even when the original threat disappears, demonstrating a form of transgenerational memory comparable to an institutional knowledge base. Agricultural scientists now apply these principles through priming techniques that expose crop seedlings to mild stresses to enhance hardiness in subsequent generations. Evolution's experimental approach stands beyond biology into geochemical processes. It's a great oxidation event when photosynthetic bacteria flooded Earth's atmosphere with toxic oxygen 2.4 billion years ago represented nature's first planetary-scale chemistry experiment, one that nearly extinguished most existing anaerobic life before giving rise to aerobic organisms. Today, similar disruptive innovation continues in places like Chile's Atacama Desert, where hyperstress-tolerant microbes are experimenting with novel survival strategies under conditions analogous to Mars. Astrobiologists consider these extremophiles living prototypes for potential extraterrestrial life forms. Humanity now stands at a crossroads where we can either ignore or learn nature's experimental wisdom. Biomimetic technologies already demonstrate the power of this approach. Artificial neural networks modeled on slime mold decision-making seal feeling, concrete embedded with bacterial spores that secrete limestone cancer therapies that co-opt immune system somatic hypermutation principles. The emerging field of evolutionary robotics takes inspiration further, creating machines that autonomously test and refine their own designs through simulated natural selection. Swiss researchers recently demonstrated this with evolutionary drones that optimize their flight stability through iterative crashes, a mechanical parallel to biological trial and error adaptation. As climate change accelerates, understanding nature's experimental frameworks becomes existential. Coral reefs display both the promise and peril of this perspective. While some species succumb to bleaching, others reveal unexpected resilience through symbiotic partner switching or epigenetic regulation of heat shock proteins. The solutions? Two anthropogenic challenges may already exist in nature's vast library of prototype adaptations, waiting to be discovered through careful observation. Ultimately, every living organism represents a successful experiment among countless failed trials across deep time. The creatures we see today are nature's validated hypotheses, proof of concepts that endured rigorous environmental testing. In recognizing life as history's longest-running innovation lab, we gain not just engineering insights, but profound respect for the creative destruction that shapes our world. The question now is whether humanity will study these eternal scientists' methods before our own uncontrolled experiments rendered their laboratories extinct in the intricate dance of life. Nature reveals itself as a master of energy and information, employing principles of quantum mechanics in ways that astound and inspire. The world of biology is not merely a collection of organisms competing for survival. It is a sophisticated network of interactions where the laws of physics and chemistry intertwine to create a tapestry of life that thrives against all odds. This hidden dimension of life showcases the remarkable adaptability and ingenuity of organisms that have evolved to harness the very fabric of the universe. Consider the phenomenon of photosynthesis, a process that seems deceptively simple, yet is underpinned by complex quantum mechanics. In the chloroplasts of plants, light energy is captured and converted into chemical energy with astonishing efficiency. Researchers have discovered that during this process, electrons behave in a manner reminiscent of quantum particles existing in multiple states simultaneously. This quantum coherence allows plants to transfer energy through their photosynthetic systems with an efficiency that far surpasses our best solar panels. The ability of plants to utilize quantum effects to optimize energy transfer is a testament to the evolutionary innovations that have unfolded over millions of years, revealing a level of sophistication that challenges our understanding of energy conversion in the depths of the ocean. Electric fish, like the elephant nose fish, navigate their murky environments using a remarkable ability to generate and detect electric fields. These fish emit weak electrical pulses that create a three-dimensional map of their surroundings, allowing them to identify objects and prey based on the distortions in the electric field. This active electrolocation system is akin to advanced radar technology, showcasing how evolution has crafted intricate sensory mechanisms that enable survival in challenging habitats. The precision with which these fish can interpret electrical signals demonstrates nature's capacity to exploit the fundamental forces of physics for practical purposes. Birds, too, exhibit extraordinary adaptations that harness the Earth's magnetic fields for navigation. The European robin, for instance, possesses specialized proteins in its eyes that allow it to perceive magnetic fields through quantum entanglement. This biological compass enables the bird to migrate thousands of miles with remarkable accuracy, relying on a mechanism that scientists are only beginning to understand. The interplay between biology and quantum physics in this context highlights the intricate connections that exist within the natural world, where organisms have evolved to utilize the very forces that govern the universe. The energy conversion capabilities of deep-sea organisms further illustrate nature's mastery over energy and information. Giant tube worms, thriving near hydrothermal vents, rely on symbiotic relationships with chemosynthetic bacteria to convert toxic hydrogen sulfide into organic compounds. This process allows them to flourish in complete darkness, demonstrating how life can adapt to extreme conditions by harnessing the energy available in its environment. The efficiency of these chemosynthetic pathways has inspired new industrial methods for utilizing waste products, showcasing how nature's innovations can inform human technology. At the microscopic level, the molecular machinery of life operates with astonishing precision. ATP synthase, the enzyme responsible for producing adenosine triphosphate ATP, functions as a molecular motor that converts proton flow into chemical energy. This rotary mechanism achieves near-perfect efficiency, illustrating how evolution has fine-tuned cellular processes to maximize energy production. The principles underlying ATP synthase are now being explored for applications in nanotechnology and energy storage, revealing the potential for nature's designs to inform our technological advancements. As we delve deeper into the realm of quantum biology, we uncover a wealth of knowledge that challenges our understanding of life itself. The ability of organisms to exploit quantum phenomena for survival raises profound questions about the nature of consciousness, perception, and the interconnectedness of all living things. The discoveries in this field suggest that life is not merely a series of biochemical reactions, but a complex interplay of energy and information that transcends traditional boundaries in a world grappling with environmental challenges and energy crises. The lessons learned from nature's quantum biologists offer a glimmer of hope. By studying the intricate mechanisms that have evolved over billions of years, we can glean insights into sustainable practices and innovative technologies that align with the principles of the natural world. Potential for biomimicry and the application of quantum principles in engineering and energy production could pave the way for a more harmonious coexistence with our environment. Ultimately, the exploration of quantum biology invites us to reconsider our place within the tapestry of life. As we uncover the secrets of energy and information that underpin the natural world, we are reminded of the profound interconnectedness that binds all living organisms. Nature, in its infinite wisdom, has been conducting experiments for eons, revealing the elegance and complexity of life in ways that continue to inspire and challenge our understanding. In embracing the lessons of these quantum biologists, we can forge a path toward a future that honors the intricate relationships that sustain us and the planet we call home. In the grand narrative of life on Earth, resilience emerges as a defining trait, manifesting in the remarkable structures and systems that organisms create to withstand the trials of their environments. Nature's architects, from the tiniest microorganisms to the largest mammals, have evolved ingenious strategies to build and adapt, crafting habitats that not only ensure their survival, but also foster the flourishing of entire ecosystems. These resilient builders demonstrate that the ability to innovate and adapt is as crucial as the strength to endure, revealing a profound understanding of the interconnectedness of life. Take, for instance, the intricate nests of the weaver bird, a master craftsman of the avian world. These birds construct elaborate hanging nests woven from grass and twigs, often suspended from the branches of trees to protect their young from predators. The weaver bird's ability to manipulate materials with precision and creativity showcases an innate understanding of structural integrity and environmental challenges. Each nest is a testament to the bird's resilience, built to withstand harsh weather conditions and the scrutiny of potential threats. The communal aspect of nest building, where multiple birds collaborate to create a single structure, further emphasizes the importance of cooperation in nature's architectural endeavors. In the underwater realm, coral reefs stand as monumental testaments to resilience and collaboration. These vibrant ecosystems, formed by the calcium carbonate skeletons of tiny coral polyps, provide shelter and sustenance for countless marine species. The process of rebuilding is a delicate balance of growth and decay, where corals continuously secrete calcium carbonate while also responding to environmental stressors, such as temperature fluctuations and ocean acidification. The symbiotic relationship between corals and zooxanthellae algae is crucial. The algae provide energy through photosynthesis, while the corals offer protection and nutrients. This mutualistic partnership exemplifies how resilience is not merely an individual trait, but a collective effort that sustains entire communities on land. The resilience of trees is evident in their ability to adapt to changing climates and environmental conditions. The ancient bristlecone pine, for example, can live for thousands of years, enduring harsh winds, extreme temperatures, and limited water availability. These trees have developed unique adaptations, such as thick bark, to insulate against cold and deep root systems to access groundwater. Their longevity and ability to thrive in inhospitable environments serve as a reminder of nature's capacity for endurance and innovation. The rings of a bristlecone pine tell a story of resilience, each layer representing a year of survival against the odds. In the realm of microorganisms, the resilience of bacteria and fungi is nothing short of extraordinary. These tiny architects play a crucial role in nutrient cycling and soil formation, creating the very foundation upon which ecosystems thrive. Mycorrhiza fungi, for instance, form symbiotic relationships with plant roots, extending their networks to access water and nutrients while enhancing soil health. This underground collaboration fosters resilience in plant communities, allowing them to withstand droughts and other environmental stresses. The ability of these microorganisms to adapt and thrive in diverse conditions underscores the importance of their role as nature's builders, shaping the landscape and supporting life above ground. The resilience of nature's architects extends to the animal kingdom as well. Termites, often underestimated, are remarkable builders that create complex mounds and colonies that regulate temperature and humidity. These structures, made from soil, saliva, and feces, serve as microhabitats for various organisms and play a vital role in nutrient cycling. The intricate design of termite mounds, with their ventilation systems and chambers, showcases an understanding of environmental engineering that rivals human construction. The ability of termites to adapt their building strategies in response to changing conditions exemplifies resilience inherent in nature's designs. As we explore the resilience of these architects, we're reminded of the lessons they impart in a world facing unprecedented challenges. From climate change to habitat destruction, the ability to adapt and innovate is more crucial than ever. Nature's builders demonstrate that resilience is not merely about survival. It is about thriving in the face of adversity and creating systems that support life in all its forms. By studying these remarkable organisms and their strategies, we can glean insights into sustainable practices that honor the interconnectedness of life. In our quest for a more sustainable future, we must embrace the principles of resilience that nature has perfected over millions of years. By learning from the architects of the natural world, we can develop solutions that not only address immediate challenges, but also foster long-term sustainability. The intricate designs of weaver birds, the vibrant ecosystems of coral reefs, the enduring strength of ancient trees, and the collaborative efforts of microorganisms all serve as blueprints for a future where humanity coexists harmoniously with the environment. Ultimately, the story of nature's resilient architects is a testament to the power of innovation, cooperation, and adaptability. As we navigate the complexities of our modern world, we must look to these builders for inspiration, recognizing that the path to a sustainable future lies in our ability to learn from the resilience that has shaped life on Earth for eons. In embracing the lessons of nature's architects, we can forge a new narrative, one that honors the interconnectedness of all living things and celebrates the enduring spirit of resilience that defines life itself. Beneath the surface of seemingly static ecosystems, an invisible army of organisms wages a quiet revolution, transforming environments and shaping the planet in ways we're only beginning to understand. These are nature's ultimate changemakers, microbes, fungi, and unassuming plants that quietly but relentlessly alter their surroundings, creating conditions favorable for life where none existed before. Their work happens on timescales ranging from minutes to millennia, their impact visible not in sudden upheavals, but in the gradual transformation of worlds. The pioneering lichens demonstrate this transformative power with elegant simplicity. These composite organisms, part fungus, part algae, or cyanobacteria, literally create soil from bare rock through biochemical warfare. Their fungal components secrete acids that slowly dissolve stone, while photosynthetic partners generate energy to fuel the process. Lichens are Earth's original terraformers, breaking down mountains into fertile ground one molecule at a time. In the Arctic tundra, they've been clocked creating soil at a rate of 1 millimeter per century, slow by human standards, but unstoppable over geological time. Their patient work laid the foundation for all terrestrial ecosystems, transforming a barren rock planet into a world capable of supporting forests, grasslands, and ultimately human civilization. Earthworms perform similarly heroic work beneath our feet, their intestinal alchemy converting dead plant matter into fertile soil. Charles Darwin calculated that in healthy English pastures, worms move 10 to 15 tons of soil per acre annually, completely recycling the topsoil every few decades. But recent research reveals even more surprising capabilities. Earthworms actively reshape microbial communities in soil, favoring beneficial bacteria that suppress plant pathogens while enhancing nutrient cycling. Their tunneling creates pore networks that allow soils to absorb rainfall up to 10 times faster than compacted earth, reducing flooding while recharging aquifers. Urban ecologists now use worm-based solutions to rehabilitate degraded city soils, proving these humble invertebrates remain essential partners in creating fertile ground, literally and metaphorically. The plant kingdom's quiet revolutionaries include certain tree species that actively engineer their own ecosystems. The Alaskan yellow cedar Calotropis nucatensis is particularly remarkable. Its fallen needles acidify the soil to levels toxic to competing plants while simultaneously promoting beneficial mycorrhizal fungi. Over centuries, this creates cathedral-like cedar groves where little else grows. Even more dramatically, the African acacia species Vachellia drepanalobium manages entire savanna landscapes through an intricate web of alliances and defenses. These trees maintain standing armies of symbiotic ants housed in swollen thorns, paying them with nectar rewards to fiercely defend against herbivores. The ants' territorial behavior shapes grazing patterns across whole ecosystems, demonstrating how a single tree species can transform landscapes through biological diplomacy. Microorganisms orchestrate planetary-scale revolutions invisible to the naked eye. Cyanobacteria sparked Earth's most dramatic environmental transformation 2.4 billion years ago, when their oxygenic photosynthesis flooded the atmosphere with toxic oxygen, an event we now call the Great Oxygenation Event. Today, their modern descendants continue shaping global systems, with marine phytoplankton producing over half the world's oxygen while sequestering billions of tons of carbon. Scientists recently discovered that certain bacteria in Arctic permafrost are actively preventing methane release by metabolizing greenhouse gases before they reach the atmosphere. Critical buffering system against climate change we never knew existed until now. Even seemingly destructive organisms play crucial revolutionary roles. Bark beetles, typically viewed as forest pests, actually drive essential ecological renewal. Their population surges kill weak trees, making room for new growth while creating snags that house over 40% of forest-dwelling species. Similarly, elephants in African savannas act as lumberjacks and landscape architects combined. Their feeding habits prevent forest encroachment, maintaining grassland ecosystems that support countless species. In knocking over trees, they create microhabitats for smaller organisms while releasing buried nutrients. These destructive agents are actually masterpreneurs, preventing ecosystem stagnation through carefully balanced disturbance regimes perfected over millennia. The implications for human society are profound. Tending these natural change agents offer solutions to pressing environmental challenges, Cure remediation projects employ fungi to clean oil spills and detoxify industrial waste sites. Permaculture designs mimic the soil building strategies of forests to restore degraded farmland. Cities are beginning to harness the water management abilities of wetland plants to create sponge-like urban landscapes, resilient to flooding. In every case, we're learning to work with nature's revolutionary forces rather than against them. What unites these diverse changemakers is their ability to work with the tools at hand, transforming limitations into opportunities. Alike needs no bulldozer to move mountains, just time and the relentless application of simple chemistry. An earthworm requires no plow to cultivate soil, only its own digestive system and constant movement. These organisms teach us that true revolution doesn't require brute force, but rather patience, adaptability, and the wisdom to leverage small actions into large impacts over time as humanity faces the urgent need to reinvent our relationship with the planet. These silent revolutionaries offer both inspiration and practical solutions. They demonstrate how lasting change occurs through countless small acts of transformation woven together over time. In their patient, deliberate work, we find a model for our own efforts to build a more sustainable world. One where humans become not dominators of nature, but partners in Earth's ongoing story of renewal and transformation. Perhaps most importantly, these organisms reveal that resilience and revolution aren't opposing forces, but complementary aspects of life's creative power. A coral reef recovers from hurricanes even as it slowly transforms ocean chemistry. A forest both resists change through sturdy trunks and drives change through fallen leaves, becoming soil. In this delicate balance between stability and transformation, we glimpse the fundamental rhythm that sustained life for billions of years, and may yet guide humanity toward a more harmonious future on this ever-changing planet. Beneath the surface of every ecosystem lies a living library where organisms encode, preserve, and transmit knowledge across generations in ways that challenge our very definitions of memory, learning, and intelligence. These biological archivers don't merely store information chemically, they actively curate survival strategies, environmental adaptations, and collective wisdom through sophisticated systems that make human record-keeping seem crude by comparison. Consider the monumental memory banks of ancient trees. The 80,000-year-old Pando aspen colony in Utah remembers ice ages through a sprawling root system, a single genetic individual spread across 106 acres, its trembling leaves whispering shared history through underground neural-like connections. Dendrochronologists decode centuries of climate data in growth rings with astonishing precision. A single bristlecone pine named Methuselah carries an uninterrupted 4,853-year meteorological record in its gnarled trunk. But trees don't just record time, they actively use this stored knowledge. Research shows forests experiencing periodic droughts grow denser wood in subsequent years, a form of arboreal lesson-keeping that strengthens future resilience. As phenomenon scientists term hydraulic memory. Animal memories extend beyond individual lifespans in astonishing ways. Humpback whales have maintained complex song patterns across 30 generations, with distinct cultural traditions separating populations. Pacific groups innovate melodic themes that Atlantic whales later adopt, creating a transoceanic exchange of cetacean culture. Similarly, chimpanzee troops preserve two-use techniques for millennia. The nutcracking skills observed today in West Africa's Thai forest mirror archaeological evidence from 4,300-year-old chimpanzee kitchen sites. These enduring cultural memories rival early human tribal knowledge systems in their sophistication and transmission, accuracy, revelations, shaking anthropology's human CEPTIONALISM. At the microscopic level, bacteria maintain epigenetic memories that border on clairvoyance. Quail encounters antibiotics, It doesn't just develop resistance. It remembers the threat for up to 100 generations through chemical markers on its DNA, priming descendants for rapid response. Some soil bacteria form biomineral time capsules, endospores, preserving working genes from their ancestors in crystalline matrices for centuries, ready to reactivate when needed. Virologists recently discovered similar ancient viral DNA embedded in human genomes. Junk DNA, once considered evolutionary debris, now appears to function as an immunological archive, helping recognize recurring pathogens. Plants demonstrate ontogenetic memory that puts machine learning to shame. Venus flytraps count touch stimuli before closing, remembering previous contacts to avoid false alarms from raindrops while ensuring prey is securely captured. More remarkably, wounded sagebrush plants release volatile compounds that teach unwounded neighbors to bolster defenses, with the memory of this warning lasting the plant's entire lifespan. Studies show receiver plants not only prepare for herbivores, but pass the alert to third-generation seedlings through epigenetic changes, a botanical version of ancestral trauma memory with profound implications for understanding intergenerational adaptation. The mechanisms underlying these biological memory systems are revolutionizing data storage concepts. DNA-based archiving already allows, quote, researchers to store entire movies and operating systems in bacteria genomes. A tablespoon of E. coli could theoretically hold all human-generated data. But nature's true innovation lies in its dynamic, living memory systems. The slime mold's ability to solve maze puzzles by retaining chemical memories of nutrient paths has inspired neuromorphic computing architectures that process information through adaptive resistance changes, similar to how human neurons strengthen frequently used pathways. Climate scientists now read ecological memories encoded in unexpected places. Whale earwax plugs archive marine toxin levels like tree rings, track droughts, while centuries-old coral skeletons preserve ocean circulation patterns in their calcium carbonate lattice. Perhaps most remarkably, analysis of 2-million-year-old permafrost DNA has recently reconstructed entire vanished ecosystems from microscopic genetic fragments, proving Earth itself remembers even when surface evidence disappears. These biological archives challenge modern society's obsession with externalized digital storage. The natural world teaches us that the most resilient memories are distributed, redundant, and embedded within living systems. Lessons urgently needed in our era of fragile cloud servers and disappearing cultural knowledge, as genomic research revealed how ancestral experiences shape organisms generations later. We're forced to reconsider how humanity's own biological and cultural memories influence our collective future. The memory keeper's greatest lesson may be that intelligence doesn't require centralized brains. It emerges from systems that record, process, and implement lessons across time and space, whether through fungal networks, recalling decades-old nutrient flows, or migratory birds inheriting celestial navigation maps refined over ice ages. As synthetic biologists work to engineer living hard drives and neuromorphic computers, they increasingly look to these organic archivists for inspiration, recognizing that nature's remembering systems have been perfected over eons of evolutionary debugging. Looking ahead, decoding biological memory systems could unlock breakthroughs in medicine, data storage, and climate adaptations, from drought-resistant crops bred to remember past survival strategies to neural implants mimicking the brain's molecular recall mechanisms. But beyond utility, these organisms remind us of our own place within Earth's great continuum of remembering and reinvention. In their silent archives lie not just survival strategies, but the very patterns of persistence that have allowed life to thrive through countless planetary changes. Wisdom we may need to heed in our own uncertain future. Each trembling aspen leaf, each whale song's echoing refrain, each microbial spore waiting patiently to revive, these are living testaments to time's passage, reminding us that all life exists as both student and scribe in nature's grand, unending chronicle. The question remains whether humanity will learn to read these memories and, more crucially, to respect and preserve the keepers who maintain them under the canopy of ancient forests and along the shorelines where tides have rhythmically risen and fallen for millennia. Nature has been patiently conducting its masterclass on sustainability, a curriculum refined through 3.7 billion years of trial and error. These are not passive environments, but active instructors, demonstrating principles of circular economies, renewable energy systems, and regenerative design that put human technology to shame. To walk through these living classrooms is to receive an education in survival that transcends language and textbooks, one encoded in root systems that share nutrients without competition, predator-prey relationships that maintain perfect balance, and in waste cycles where every byproduct becomes valuable currency. Perhaps no instructors are more eloquent than the old-growth forests. These venerable ecosystems operate on principles that make human economic systems seem primitive. By comparison, their circular economy begins with photosynthesis, the original solar technology, where trees convert sunlight into chemical energy with nearly 100% efficiency, dwarfing our best photovoltaic panels that struggle to surpass 30% its energy. Cascades through food webs where nothing is wasted. Fallen leaves become fungal networks currency. Decaying logs nurse new seedlings, and even dead trees continue contributing as nurse logs that may sustain life for centuries after their demise. When researchers calculated the economic value of services forests provide, from oxygen production to water filtration, they found a single hectare of undisturbed Amazon rainforest performs $6,000 worth of ecological services annually. Free of charge. Water systems teach equally profound lessons about resilience and adaptation. The intricate branching patterns of river deltas and lightning bolts follow the same fractal mathematics as our circulatory systems, demonstrating nature's universal principles for efficient distribution networks. Mangrove forests, with their stilt-like roots, demonstrate how to build flood-resilient infrastructure, while simultaneously creating nurseries for fisheries and carbon sinks that outperform terrestrial forests pound for pound. Modern engineers now mimic these blue-green designs in sponge cities that absorb stormwater like wetlands and bioswales that purify runoff while beautifying urban landscapes, proving nature solutions can be adapted even to built environments. The waste management systems operating in healthy ecosystems shame our linear take-make-dispose industrial model. In nature's classroom, there is no way to throw things. Every output becomes some organism's input. Dung beetles transform waste into fertile soil. Fungi digest petroleum spills. And even extreme environments, like acidic mine drainage, become habitats for iron-oxidizing bacteria that detoxify water while creating surreal, living artworks of orange bacterial filaments. Our most advanced recycling facilities achieve 70% material recovery on good days. The average forest recycles 100% of its biomatter while producing oxygen as a byproduct. Energy systems in the natural world operate on principles of abundance within limits, solar-powered, distributed, and resilient. Coral reefs manage to support greater biomass density than any human city while using only the energy provided by sunlight and currents. Migratory species like monarch butterflies and Arctic terns harness atmospheric patterns for efficient transportation. That makes our jet fuel dependence seem archaic. The implications for renewable energy design are profound. Wind farms now mimic school of fish formations to optimize airflow, while solar panels incorporate leaf-inspired nanotreatments that maintain efficiency even when coated with dust. Perhaps most crucially, these natural systems teach us about time horizons, where human planning struggles beyond quarterly reports or electoral cycles. Oak trees plan centuries ahead, dropping acorns that may only germinate when conditions are perfect decades later. Codorizal networks demonstrate how to invest in shared infrastructure whose benefits accrue over generations. Glacier melt patterns, tracked by lichen growth, remind us that some changes unfold across timescales. Our fast-paced culture struggles to perceive as humanity faces unprecedented environmental challenges. These silent teachers offer not just inspiration, but practical blueprints. Biomimicry specialists are translating nature's lessons into human applications, building materials grown from mycelium that self-repair like living tissue water filtration systems modeled on mangrove route, filtration closed-loop manufacturing systems, where waste equals food cities designed to function like forests, producing clean air, water, and energies. The emerging field of regenerative design goes beyond sustainability's damage limitation to actively restore and improve environments. Much like a healthy wetland naturally purifies water while creating habitat, this paradigm shift recognizes that human systems must move from exploiting nature to learning from its 3.7 billion year research and development program beyond technology. These living systems offer something even more valuable, a reconnection to the ecological consciousness that shaped our species' development. Indigenous knowledge systems preserving these lessons, from Aboriginal fire management practices to terra preta soil cultivation, demonstrate how human cultures once thrived by attending to nature's curriculum. Rediscovering this wisdom may prove essential for navigating the Anthropocene. The silent teachers don't speak in words, but in patterns, processes, and relationships written into every ecosystem. Their lessons remind us that true sustainability isn't about sacrifice or limitation, but about aligning with the abundant, cyclical flows that have sustained life since its origins. As students of these oldest professors, we're just beginning to translate their ancient wisdom into forms that can guide humanity toward a flourishing future. The classroom has always been open. Our task is learning to see, understand, and embody its timeless lessons beneath the apparent stillness of a forest floor or the quiet depths of ocean trenches unfolds the most profound performance of all, the never-ending cycle where endings become beginnings, and death fuels life in perfect symbiosis. Far from being morbid, nature's embrace of mortality emerges as its most creative act, a continuous rebirth that has transformed fallen giants into fertile soils, extinction events into evolutionary leaps, and individual sacrifices into collective survival. This is the realm of decomposers and recyclers, of ecological succession and evolutionary adaptation, where every ending carries the promise of transformation. Consider the humble log rotting in a temperate rainforest. What appears as decay is actually a spectacular rebirth in motion. As fungi, bacteria, and invertebrates break down the wood, they create a nutritional feast for plants, a nursery for seedlings, and habitat for countless organisms in the forest's understory. Scientists studying nurse logs have discovered astonishing levels of biodiversity harbored within a single decaying tree. Up to 40% of a forest's vertebrates and invertebrates may depend on dead wood at some point in their life cycles. But the real magic lies in the chemistry, as decomposers like oyster mushrooms break lignin and cellulose into simpler compounds. They release nutrients once locked in tough fibers, effectively cooking a slow-release fertilizer that will feed the next generation of trees. The process continues for decades until the log's entire mass becomes forest floor, its essence dispersed through new life in an unbroken chain stretching back to the first forests. 385 million years ago, the ocean's death transformation system operates on a grander scale still. When a whale dies and sinks to the abyssal plain, its carcass, called a whale fall, becomes the foundation for entire ephemeral ecosystems. In the initial mobile scavenger phase, hagfish and sharks strip flesh in a frenzy lasting months. Then the enrichment opportunist phase begins, with crustaceans, mollusks, and worms colonizing the bones in densities 30,000 times higher than surrounding seafloor. Finally, Sulfur-loving bacteria slowly consumed the remaining lipids in the skeletal remains over decades. A single whale fall provides as much nutrition to the deep sea as 2,000 years of normal organic rain from surface waters. These rare biological windfalls maintain species that otherwise couldn't survive in the deep ocean's food deserts. Even more remarkably, research suggests whale falls act as evolutionary stepping stones, allowing species to disperse between hydrothermal vent habitats across vast stretches of inhospitable abyss. Microbial decomposers showcase death's transformative power at the molecular level. Streptomyces bacteria, the source of most clinical antibiotics, develop their medicinal chemistry specifically to wage biochemical warfare against competing microbes in the soil's nutrient battlefield. When resources grow scarce, these bacteria initiate programmed cell death in parts of their colonies, harvesting their own kin as nutrients while redistributing antibiotics to suppress competitors. This macabre but effective strategy keeps soil ecosystems balanced while gifting humanity with life-saving medicines. It's a powerful reminder that what we call destruction often serves higher creative purposes in nature's economy, even mass extinctions. Nature's greatest tragedies paradoxically drive its most creative renewals. The Permian Triassic extinction that wiped out 90% of marine species simultaneously cleared ecological space for the first dinosaurs to evolve. The asteroid impact that ended the Cretaceous triggered the rapid diversification of mammals. Modern coral reefs continually demonstrate this paradoxical resilience when bleaching events or storms devastate sections. The blank canvas of dead carbonate structures provides ideal settlement sites for new coral recruits, often increasing biodiversity through what ecologists call the intermediate disturbance hypothesis. Death here functions not as failure, but as nature's pruning shears, a painful but necessary process that keeps ecosystems dynamic and adaptable. Human cultures, inheriting these natural rhythms, built regenerative practices, now being rediscovered. The Native American Three Sisters agricultural system, where corn provides structure for beans to climb, while squash leaves shade out weeds, mimics the layered decomposition cycles of forest succession cycles. Scandinavian farmers buried fish under crop rows, unconsciously replicating the marine nutrient cycling that feeds coastal redwoods. Contemporary regenerative agriculture now formalizes these practices, using crop rotations and planned grazing to rebuild soil, organic matter at rates matching or exceeding natural accumulation. The most profound lesson from nature's cyclical wisdom may be its value systems, where waste doesn't exist. Scarcity breeds creativity, and limitations spark innovation, where human systems see linear progression from resource to refuse. Natural systems recognize the spiral dance where outputs become inputs in an upward cycle of increasing complexity and resilience. When a mayfly dies after its single day of airborne life, its body fertilizes the river that will nourish its offspring. When a volcano annihilates entire landscapes, its mineral-rich ash creates the world's most fertile soils. The pattern repeats across scales: death and renewal, not as opposites, but as two faces of the same creative force. As humanity faces ecological crises born from our linear, extractive thinking, these natural cycles offer both warning and hope. Climate change reminds us that accumulated waste, whether carbon or plastic, eventually disrupts balanced systems. But nature's infinite cycle also demonstrates that regeneration is always possible when we work with these fundamental rhythms rather than against them. From fungal remediation of oil spills to tree planting initiatives that deliberately mimic natural forest succession, we're slowly learning to participate consciously in Earth's eternal renewal dance. Perhaps this final lesson resonates most deeply by embracing our role as both participants and custodians in nature's cycles. Humanity might yet discover our own sustainable niche in the planet's living systems. Just as fallen leaves nourish future forests and dead stars seed new solar systems, each ending carries within it the potential for transformation if we have the wisdom to recognize it. The choice before us isn't whether to participate in these cycles, we already do, but whether we'll do so consciously, respectfully, and creatively, like all successful species before us. For in nature's grand symphony, life and death are but alternating movements in an eternal opus of renewal, standing sentinel across epochs. Certain organisms serve as Earth's living archives, biological time capsules, preserving records of planetary change in their tissues, growth patterns, in very DNA. These are not merely ancient survivors, but active participants in an unbroken dialogue between past and present. Cicada, their bodies encoding climatic shifts, geological upheavals, and evolutionary turning points with astonishing fidelity. To study them is to open a living scrapbook of Earth's history, written not in stone but in sap and sinew, offering insights no fossil or ice core could provide alone. The bristlecone pines of California's White Mountains stand as the planet's oldest non-clonal organisms, with individual trees like Methuselah dating back 4,854 years. These gnarled sentinels have weathered volcanic eruptions, the rise and fall of civilizations, climatic fluctuations, while continuing to add annual growth rings thinner than a human hair. Astrochronologists have stitched together their overlapping ring patterns into an 11,000-year climate recorder timeline, so precise it can date archaeological artifacts better than carbon-14 methods. But beyond being passive recorders, keys, pines actively adapt their growth strategies across century-shifting resin production, altering needle clusters and adjusting root architectures in response to multi-generational climate patterns. Their twisted forms embody what scientists call long-term phenotypic plasticity, the ability to modify their physiology across timescales no human researcher could hope to observe firsthand. Beneath the waves, glass sponges, hexactinellida, construct skeletal frameworks of silica that persist for millennia, their intricate latticework serving as both habitat and historical archive. Colonies in Antarctica's raw sea have been carbon dated at over 10,000 years old, their glass spicules capturing traces of ancient seawater chemistry like biological hard drives. Even more remarkably, these sponges demonstrate what marine biologists term ecological memory. Their filtration systems retain optimal configurations for surviving past climatic events, which they reactivate when similar conditions recur. The recent discovery that these sponges transmit light through their silica skeletons via natural fiber optics, potentially using it to coordinate feeding across their massive bodies, adds another dimension to their role as living chronicles of deep time. Microscopic timekeepers exist as well. Permafrost bacteria like Carnobacterium pleistocenium have revived after 32,000 years of suspended animation. It's preserved DNA offering snapshots of Ice Age microbial communities. Even more astonishing, scientists successfully activated 100-million-year-old microbes recovered from deep seafloor sediments, organisms that predate the dinosaurs and remember biochemical pathways from when continents still clustered together. These Lazarus microbes don't just survive, they actively repair their DNA during dormancy, maintaining genomic integrity across geological epochs through mechanisms we're only beginning to understand. Their endurance suggests microbial memory may persist on billion-year timescales, fundamentally altering our understanding of evolutionary timelines, animal migrations, and code temporal knowledge in their very roots. Arctic terns making their 25,000-mile annual journey follow flight paths refined since the last ice age. Their navigational maps pass down through generations as both genetic programming and learned behavior. Similarly, green sea turtles return to nest on the same beaches where they hatched decades earlier, guided by magnetic maps imprinted during their embryonic development. living witnesses to coastlines that may have since eroded or shifted. Conservationists now recognize these migratory pathways as temporal corridors, biological threads connecting ecosystems across both space and time, whose disruption severs more than just movement patterns, but an unbroken chain of inherited experience stretching back millennia. Perhaps most unexpectedly, viruses serve as prolific historians at the molecular level. Endogenous retroviruses have inserted their genetic material into host DNA for eons, creating a genomic fossil record of ancient infections. Scientists recently reconstructed a 700-million-year-old viral protein from these embedded sequences, discovery that's revolutionizing our understanding of both virology and evolutionary developmental biology. Even more remarkably, bacteriophages, viruses that infect bacteria preserved in Siberian permafrost, have yielded viable 30,000-year-old viral DNA, not as dormant particles, but as fully functional genes that immediately began replicating when introduced to modern bacterial hosts. These viral time capsules demonstrate that genetic memory can persist across epochs with near-perfect fidelity under the right conditions. The practical applications of studying these biological historians are profound. Ancient tree rings help refine climate models by providing ground truth data for prehistoric atmospheric conditions. Sponge silica deposits offer baseline measurements for ocean acidification trends predating human industrialization. Revived microbes provide novel enzymes for biotechnology, including a 120,000-year-old ice-bound bacterium that produces an antifreeze protein, now used to preserve transplant organs. Perhaps most crucially, understanding how these organisms preserve information across deep time may help humanity to safeguard biodiversity against accelerating environmental change. Yet, beyond utilitarian benefits, these eternal witnesses offer something more profound, a visceral connection to deep time that no textbook or fossil display can match. To stand before a bristlecone pine that was already ancient when the pyramids were built is to experience what environmental philosopher Kathleen Dean Moore calls time-hauntedness, the shattering realization that we are brief participants in stories much larger than ourselves. To sequence DNA from a resurrected bacterium is to hold a living relic from when woolly mammoths still walked the earth. These organisms collapse the artificial divide between past and present, reminding us that all life exists along an unbroken continuum stretching back to Earth's primordial beginnings as humanity grapples with the Anthropocene's disruptions. These timekeepers deliver an urgent message written in growth rings and genetic code. The planetary systems we take for granted have undergone radical transformations before, and life has always adapted, but never without cost, and never with guarantees. Their persistence through previous upheavals offers both warning and hope, evidence of nature's resilience, alongside sobering proof that recovery operates on timescales dwarfing human lifespans. In the end, these eternal witnesses teach us that memory is not just stored, it is lived. The past persists not as artifact, but as active participant in the present moment, in the seasonal rhythms of ancient trees, the migratory instincts of ocean-crossing birds, and the resurrected metabolisms of microbes that remember when the world was young. To heed their lessons is to recognize our own role as both inheritors and custodians of deep time, temporary stewards of biological memories that will outlast us by millennia, and humble students of Earth's oldest living historians in hidden laboratories unseen by the naked eye. Nature's molecular engineers perform daily miracles of transmutation that shame our most advanced chemical plants. These are not primitive reactions, but sophisticated biochemical factories, coned across millennia, where leaves forge sunlight into sugar, bacteria spin waste into gold, and simple organisms routinely accomplish what human scientists still struggle to replicate. Welcome to Earth's oldest and most efficient chemical plants. It's operating since life's dawn, where every organism serves as both chemist and laboratory, transforming base elements into living matter through processes we're only beginning to understand. The humble leaf contains the most refined solar chemical plant in existence. The chloroplasts within plant cells perform photosynthesis with quantum-level precision. Their pigment molecules arranged in perfect arrays to capture 90% of incoming photons. This light energy splits water molecules a reaction chemists can only accomplish with massive electrical inputs, liberating oxygen that shaped our atmosphere over billions of years while channeling hydrogen into sugar production. The entire process occurs at room temperature, using non-toxic manganese catalysts rather than the rare metals our industrial processes require. What's more, plants self-repair their photosynthetic machinery constantly. A single square meter of leaf surface replaces its entire complement of proteins every 45 minutes. It's a feat of molecular recycling no human factory could hope to match. Scientists attempting to create artificial photosynthesis struggle to reach 10% of nature's efficiency, despite billions in research funding in cutting-edge nanomaterials. Bacteria demonstrate even more astonishing chemical virtuosity. Geobacter, species found in contaminated soils, can metabolize radioactive waste, converting uranium dissolved in groundwater into insoluble crystals that settle harmlessly out of solution. Other bacterial chemists perform alchemy in earnest. Drostonia metallodorans precipitates pure gold from toxic gold chloride solutions, while Cupriavitis metaldurans, discovered in a French gold mine, survives lethal metal concentrations by crystallizing heavy metals into nanoscale nuggets stored safely in its cells. These microbial metallurgists don't just endure poisonous environments, they thrive by transforming toxins into biologically inert forms, offering revolutionary models for bioremediation and metal recovery from industrial waste. Marine organisms produce chemistry that borders on science fiction. The bloodworm Glycera debranchiata manufactures fang tips containing crystalline copper, the only known animal to incorporate this metal into structural biology. Even more remarkably, deep-sea hydrothermal ventworms like Riftia pachyptilla thrive in complete darkness through symbiotic bacteria that chemosynthesize food from hydrogen sulfide, a process converting toxic vent emissions into life-sustaining energy. The enzymes these tube worm symbionts use to harness chemical energy inspire new biofuel technologies, proving that solutions to energy challenges may lie in organisms thriving at ocean depths we've only begun to explore. Fungal chemists rival their bacterial counterparts in sophistication. Oyster mushrooms Pleurotus astretus produce oxalic acid that dissolves petroleum hydrocarbons, making them powerful allies in cleaning oil spills while simultaneously converting the pollution into edible fungal biomass. Mycelial networks secrete extracellular enzymes that break down lignin and cellulose, nature's most recalcitrant polymers, turning fallen trees into rich soil. Industrial chemists now harness these fungal enzymes for everything from biofuel production to sustainable textiles, replacing toxic processes with biological alternatives. The human implications of these natural chemical masters are profound. Bio-inspired catalysts could revolutionize industrial chemistry using abundant metals rather than rare platinum group elements. Its enzymatic processes may replace toxic solvents in pharmaceutical manufacturing. Bacterial metal recovery methods offer sustainable alternatives to destructive mining practices. Photosynthetic microbes provide templates for carbon-neutral fuel production. Fungal cleanup crews remediate contaminated sites while producing useful biomass. Beyond practical applications, these organisms challenge our basic assumptions about chemistry's possibilities. They perform feats our laboratories can't replicate, eporating at ambient temperatures, using water as solvent, and achieving 100% atom economy where no byproducts go to waste. Their secret lies in precision engineering and nanoscale, where human chemistry relies on brute force, heat, and pressure. Nature uses perfectly shaped enzyme active sites and quantum tunneling effects to lower reaction barriers elegantly. Perhaps most importantly, biochemical maestros demonstrate that sustainability isn't a limitation but an opportunity for innovation. Each chemical transformation in nature serves multiple functions. A leaf's photosynthesis produces food while regulating atmospheric gases. A cadial colony cleans water while precipitating valuable minerals. A fungal network digests pollution while building soil fertility. In an era of climate crisis and resource depletion, these time-tested chemical strategies offer blueprints for circular economies, where waste becomes feedstock and energy comes from renewable flows. As we stand on the brink of a new biochemical revolution, one merging nanotechnology, synthetic biology, and green chemistry, we would do well to apprentice ourselves to nature's master chemists. Their solutions have been field-tested for billions of years across every environment Earth can offer. The future of chemistry may not lie in inventing new reactions, but in understanding and emulating those life has already perfected. It's working with nature's alchemical wisdom rather than against it. In their molecular artistry lies the promise of truly sustainable technology that heals rather than harms the planet we all share. The story of life on Earth is not a linear march of progress, but a vast, interconnected symphony, one that has been playing for billions of years, with each organism contributing its unique note to the grand composition. From the quantum-level precision of photosynthesis to the ancient wisdom encoded in the rings of bristlecone pines, nature reveals itself not as a collection of isolated phenomena, but as a deeply interwoven tapestry of relationships, adaptations, and ceaseless creativity. What makes this symphony so extraordinary is its resilience. Life has weathered asteroid impacts, ice ages, and catastrophic volcanic eruptions. Yet, it has always found a way to reinvent itself, not through brute force, but through collaboration, flexibility, and an unwavering commitment to balance. Forests communicate through fungal networks, carrier, trade, genetic innovations like currency, and even the smallest microbes transform toxins into nourishment. There are no wasted movements in this symphony, no disposable players, only an intricate dance of giving and receiving, of death feeding new life, of endings birthing beginnings. Now, for the first time in Earth's history, a single species holds the power to alter the course of this symphony, not just for itself, but for every other living thing. The question is no longer whether we can reshape the world, but how we will choose to do so. Will we be the dissonant note that disrupts the harmony? Or will we learn to listen to the quiet wisdom of lichens breaking stone into soil, to the mycelial networks stitching ecosystems together, to the ancient trees that remember climates long past? The answer lies not in abandoning progress, but in redefining it. Nature does not resist change, thrives on it, but it does so within boundaries, respecting the delicate equilibrium that allows life to flourish. If we are to write the next movement of this symphony, it must be with humility, curiosity, kia, and a willingness to learn from masters who have been refining their craft since life first stirred in primordial seas. The music is still playing. The choice of what comes next is ours. And somewhere in the rustling leaves, the murmuring tides, and the silent work of microbes, the earth whispers the same refrain it always has: adapt, create, endure. Good night.